Hi there, Ms. Lams here. In this video, we would like to share some IA feedback from previous years. So hopefully after you watch these videos, it will help you to remind you a few things that is common errors. Okay, so IA feedbacks. Here we go. So first part is the personal engagement. So personal engagement actually takes up about 2 points. And this criteria is actually judged on quite holistic judgment. So what things examiner is looking for example, is why you would like, first thing, why would you like to conduct this experiment? Usually this will add into the context as to why you want to do it, okay? So for example, people were saying that, they're interested with microbiology, and then the reason why they will be interested with microbiology. So for example, there's some personal experience towards it, things like that. Now, in addition to this, okay, a lot of other areas comes into play. For example, how dedicated your attitude towards this experiment. So how can an examiner judge on that is, for example, your holistic, this is judged holistically, okay? So based on your experiment design, for example, if you're very dedicated towards your IA, we expect you to have a very thorough biological research and then plan things ahead, do some pilot study, explain your steps all the way along, show your thinking process, okay? Of course, also we hope that your experiment could be quite innovative, okay? So and this will also come into play is the level of difficulty, how easy or difficult it is to obtain your data. So for example, some experiment could be quite labor intensive, okay? So maybe some experiment that could be very simple but labor intensive, or could be something that is quite challenging to, it's out of the scope of the IB syllabus, but then you can, through your research, through communication with other people, you have come up with this experiment design, things like that. So of course, experiment from the class, okay, lessons is not allowed. So what are we looking at? For example, some of the prescribed lab, okay, so IB do have a list of prescribed labs that students need to conduct. So if your IA experiment is about that, then the experiment design would not work. So generally speaking, for the personal engagement is, although it's only out of 2, but actually this one the examiner will judge holistically after reading the whole report. Okay, so moving on. Okay, so this is some of the examiner report from previous years. Okay, so when the examiner are marking it, okay, or giving some feedbacks, okay, so you can pause this video and then take a quick look at this. Okay, again, you can see that the examiner have mentioned that to mark this criteria is a holistic approach. Okay, so yep. Okay, so next part is explorations. Okay, so exploration is actually quite a heavy topic, so it's out of 6 marks. Okay, so what comes into play in explorations? Okay, so in the past, you probably, most students will say, oh, that is the introduction part, so that is the theoretical part about the biological knowledge part. So yes, of course you need to have the biological knowledge, but what comes a little bit more challenging is you need to explain your experiment steps. So in this exploration part, actually it involves several parts. Okay, so previously, okay, if you have done your mock IA, okay, then you will understand better as to how the exploration works. Sometimes students will use the mock IA as their pilot study for the real IA. Okay, so in what we're trying to say is that your pilot study is very important. So for example, it is related to your experiment design. Like we said before, you need to explain to examiner that why you're using certain concentration, or why you're using certain volume, or why you're using this method. Okay, so you need to explain. Okay, you need to explain. That means to justify along the way. Okay, of course, while you're writing the biological theory, okay, you also need to, when you set up your hypothesis, you do need to have some external experiment, study, or publication to support your hypothesis, things like that. Okay. And then now next part, according to the examiner feedback, common errors in the exploration part is the research question not detailed enough. Okay, so you need to, your research question needs to be very specific. Generally speaking, we will say that, oh, okay, so when you write down your research question, first of all, it must be in question format. Secondly, you need to mention your IV and DV. And then to be safe, we will recommend students to mention 2 CV control variables. Okay, just a reminder as well, if you're involving some of the other species, okay, generally speaking, you will, okay, then you need to mention the binomial name, okay, and put it in italics, okay, in your RQ. And then your experiment design. Now, previously we've mentioned that in the past study, it helps you to justify why you're taking certain steps or use certain concentration or volume. So what else is involved? Okay, so when you design your experiment, then we will, let's say if your IV that means your independent variables is continuous, then you need at least 5 increments plus the negative control. Okay, negative control. Okay, so generally speaking, if you have more substance added into your experiments, okay, so for example, I couldn't think of one, but for example, if you have more, then you need to add a few more than one negative control. Okay, so yes, so I can use example of the microbiology lab. So for example, students were checking whether there's some microbes in the tea itself. Then they need to do another lab that is with no microbes in it. Okay, so like things like that. Okay, so yeah. And the next one is the uncertainty for the equipments. Okay, you need to list them out. Okay, so in exploration part, you also need to list out the apparatus list and the chemical list and things like that. So when you mention about the apparatus list, the equipment part, you need to include the uncertainty. Okay, there's only one exception, is the beaker. Okay, so because we don't use the beaker's marking for measuring the volume, so therefore there will be no uncertainty for the beakers. Okay, and the next one is generally speaking, the diagrams. It depends on how many pages you have, because actually for IA there is a 12-page limit. So if you have space, okay, of course you can add some photos, okay, label photos for the experimental setup. Okay, so for example, the qualitative data. Generally speaking, if you mention the photos, okay, we will prefer students to mention about the qualitative data only. Actually, you don't need a photograph or a draw diagram for experimental setup. You don't need that. Okay, generally speaking, you don't need that. But it will be beneficial if you have a labeled photo for your qualitative data, which we will be explaining later on. Okay, so next one, also under the category of the exploration is the controlled variables. Okay, so you need to explain how you control other variables that will not, so that it will not affect the result of your experiment. So generally speaking, in IB, okay, we prefer students to think of like at least 5 controlled variables. Okay, so and as the green IA guidelines, okay, booklet have shown, okay, for the control variables, we also recommend students to use a table format. Use a table format with 3 columns. Okay, so 3 columns. One is the control items. Okay, control variables items. Okay, so and then second column is Y. Okay, so generally speaking, such as the biological reasons. Okay, so theories wise, and then how you control it. Now, there's several things that you need to watch out for. So for example, the controlled variables, you need to include the numbers that is involved. For example, if you say, oh, you need to control the temperature, let's say you control the temperature of the experiment, then you need to mention at what temperature. Okay, you need to say that, or control the temperature at, let's say, 29 degrees Celsius. Okay, things like that. And also another thing that examiner has mentioned in the examiner report is that avoid the use of the word amount, because the word amount is very unclear. Okay, and so what can we use instead? So what we can use is the word concentration or volume. Okay, so these are the good words instead of the word amount. Okay, and then regarding to how, okay, so how, again, you need to mention the numerical values. Okay, and also the equipment that you use. So for example, how do you control the temperature at 29 degrees Celsius? Then you can say that, okay, I insert a thermometer there, okay, to make sure the water bath is at 29 degrees Celsius or things like that. Okay, so so that's the controlled variables. Okay, make sure you express it in a table format with 3 columns. Okay, and the next part is the data presentations. Okay, so also again is in one of the IA guidance. Okay, so we prefer to use the SI unit. Okay, so don't use milliliters. Okay, you should be using cm cube. Okay, if it's chemistry, then probably you'll face the term dm cube. Okay, Of course, you also need to mention the steps that you need to how you can measure the IV range. Okay, so for example, in your method, basically in your method, you mention that like what volume of water you add, the mass of the powder they use to mix with it, things like that. Okay, now this is a very common one that students forgot. Okay, is the safety. Okay, safety. So a safety, 3 parts is actually in the criteria as well. So safety, ethical, problems and environmental issues. What are you looking for? So for example, it's very simple. All you need to mention is that, oh, for example, you wear your safety goggles and lab coat. In some experiment, you might need to wear some gloves as well. Ethical, so ethical such as how can you prevent to minimize the food waste, okay? And the environmental issue, how you dispose some of the used material plate, things like that. So it's very straightforward, but sometimes students, when they are in a rush, they forgot about this aspect, okay? So this is very important. As you can see in the next few slides, why it is so important. Now, next one. So, okay, so this one is actually another quote from the examiner report. Okay, you can pause the video and see, but I just want to highlight a few things. Okay, so you can pause it and see. Is that, for example, right here, they were saying that, oh, some students mentioned the argue to be how different amount of sugar effect on cell respiration yeast in bread making. Okay, so very simple. But then they say that, okay, so need to mention the species of a yeast, so that is the biological names. Okay, sugar that you use. Okay, so you should know by then when you are doing IAs, you should know that there's different types of sugar. So what is it, sucrose or fructose? What is it? Okay, and amount, they really don't like the word amount. Okay, you prefer to use the word mass, volume, something that you can quantify. Okay. So that's the idea about this part. Okay, so this actually taken from one of the previous year's examples. Okay, from the mock IA. Okay, so and I make a meme and say, okay, I knew that you didn't refer to the green IA booklet. Okay, when I see this now, what happened here is that actually the student did not separate into 3 columns. Okay, and then they just mentioned temperature. Okay, they did mention about the values. Okay, but they didn't mention how they control it. So the how is missing. Okay, the how is missing. So again, you probably wonder, oh, do I have to use the 3 columns? Usually we recommend you use the 3 columns to help you to make sure you have got all the points in. Okay, so incubator period, okay, you mentioned, like, for example, 24 hours, mention the time. Okay, to quantify it. Okay, yes, numbers. Okay, so they try to explain why, okay, but then they mention about the equipment again. So the equipment is missing as well. So yeah, so this is not a very good example. Okay, so again, so it's also in the green IA booklet. You can see on page 10. They also recommend you, okay, so why, okay, and how. Okay, so this is the key things. Okay, key things that you need to mention. Okay, again, exploration, why I was saying that, like, it's important to mention about safety, ethical, and environmental reasons. Okay, so actually IB marked it in criteria. Okay, so but you can see that, okay, for each mark score band, okay, they also have safety, ethical, and environmental issue in all categories score band. So if you miss out the safety, ethical, environmental issues, then your mark can be penalized and then drop into the next score band. Okay, so again, so make sure you mention these 3 things when you are doing your exploration part. Okay, lab safety. Okay, now another things that comes from the examiner feedback is that you need to mention how you carry your lab safety. So for example, one of the students says, okay, so they were the experiment involved UV light. Okay, so then they say that, oh, okay, don't directly look into the UV light and don't touch the ascorbic acid directly. But why and how? Okay, why you shouldn't be looking at the UV light directly? Okay, because UV light is high energy, it can damage your eyes. Okay, and then what can you do? Okay, you should use safety goggles with the filter that can block the UV light. Okay, another thing is do not touch the ascorbic acid directly. Why? Because ascorbic acid is corrosive, so you should wear your gloves or things like that. So you need to explain why and how. Okay, so actually there's a lot of things involved. Okay, so don't treat it too lightly. Okay, and the next one is the qualitative data. This partly overlap with the analysis, but we will go through a few of them. So qualitative means that's something that's non-numerical, so your observations. So generally speaking, student will take a picture of it and then leave it that way. Okay, so that's not a recommended one. So what you do is, if you take a picture, you should label your photos. Okay, so key things, add labels. Okay, and then you should have words to describe what you see. Okay, it will be beneficial if you mention the comparison between the results. So for example, the comparison between the negative control and something else. Okay, or for example, if you have a continuous variable, then for example, whether there's a trend, is it getting darker color or lighter color, things like that. And a good qualitative data is the student can refer back to the qualitative data. So that means that the qualitative data actually supports the quantitative data, the numerical data. So you need to refer your qualitative data back in your conclusion later on. Okay, so another thing is, okay, so one of the examples from previous years, so if for example they say that, okay, so the criteria for which seed are determined to have germinated is through observation of sprouting of any kind. So this student was looking at seed germination, and then they just say, oh, how you observe the data is if you see sprouting, but without any photograph to support what is meant by sprouting, is not detailed enough. Okay, so you want to have a useful, okay, and detailed enough qualitative data. Okay, yes, again, so reminder, SI unit. Okay, so again, so for example, if let's say if your experiment involves time, okay, then actually to make sure you standardize, okay, standardize the unit. So for example, let's say if you time something and then it turns out to be 1 minute and 43 seconds, okay, then you do not put 2 units in together. So you convert it all into, let's say, seconds and things like that. Okay, so you need to unify your units. Okay, so you need to be sensible as to which unit you'll be using. So for example, let's say if you know that the change is very minimum, okay, so for example, let's say the change of height in gelatin gel in the gelatin gel experiment, then you should be using mm rather than cm. So you need to be sensible with the unit that you chosen. Okay, so final checklist. Okay, so actually our IA guidance, okay, is actually very useful. The green booklet is very useful. So for example, it helps up actually this one. Okay, so this is actually taken from the syllabus as to how the examiner marked. Okay, but here is actually a more detailed, a more refined as to what you need to checklist. Okay, so make sure you checklist things like that. Okay, so yeah, so make sure you do that. And some of the examiner feedback. Okay, just one more. Okay, and then I think it's nearly the end. Background. Okay, what is needed in the background? So it must be relevant. So yes, this is very important as well. So it must be relevant to your experiment. So for example, because you are limited by the page number, 12 pages only, so you don't want to drown the examiner with a lot of irrelevant information. So you need to choose your background research carefully. Okay, so and then you need to, this is again very important, that you need to justify why you use this independent variables. A very simple way is to mention like what happens in the pilot study. Okay, so why you use certain concentration, volume, and so on. And the dependent variable needs to be explained. Now this is also another important and challenging part. So for example, how you interpret the data. Okay, so for example, let's say someone was looking at plant growth, so you look at the height over time and things like that. So you need to explain how you interpret the result related to the photosynthesis rate or things like that. Okay, to the result basically. The discussion of the control variables. Okay, so again we mentioned about using the table with 3 columns, explaining why you need to control it and how you should control it. Okay, so this is very important. Again, so this is actually some of the uncontrolled variables that should have been controlled. So for example, room temperature. So a lot of experiments conducted in room temperature, but you need to explain what is the impact. So you cannot control it, but what's the impact of it. Okay, so So this is a final checklist about the explorations. I know it's a very big one, but please note that this is just a brief list, okay? Your experiment may need a little bit more, okay? So just an idea. So let's go through them very quickly. Introductions, okay? So your context, why you want to do it, the biological theory, okay? Possibly using some external research to support it. Of course, you need to mention about the occasionally mention about the methods, okay, why you use this method, okay, what's the benefit, okay, of it. And then, of course, you need to have your research question in a question format. So we mentioned before, you need to mention your IV, DV, and 2 CV in your research question. And of course, you need your hypothesis, okay, and then the IV, DV, CV, okay, we've mentioned it in previous part. IV to justify it, you can use it with pilot study. DV, you need to explain how to interpret your experiment result with the dependent variables. CV is using in a table format. The CV items, the why you need to control it and how you can control it. Okay, and the next one is the apparatus list. Okay, so when you mention about apparatus list and the chemical list, okay, so apparatus lists do need to have the uncertainty. And of course, you mentioned about the safety, ethical, and environment. You need to elaborate a little bit as well. And then the method, of course, in numbering points, okay, in bullet points, numbering order, and the qualitative data. So you need to make it useful. What we mean by that is that, for example, you should have a trend. Your qualitative data needs to have photos and descriptions about it, okay, hopefully have a trend, so then you can use it to support your quantitative data, and you can refer it back when you are writing your conclusions. So that's about it for the personal engagement and exploration. I hope this video helps you. If you have any questions, feel free to comment in the video link below. And that's about it. So let's look forward to the next video about the analysis next time. So good luck and bye for now.